try to see if we can get. Okay, I'll see if I can actually get this to work now. This is supposed to work. Allison, can you see the BBC brain thing now?
Yes, I can.
Sweet. Okay, cool. So we can actually look at that today. Nice. That's exciting. Got that. Oh, I should probably put a little bit back to that. Okay. Sweet. Father, the Son, the Holy Spirit. Amen. God, grant me the serenity to accept the things I cannot change, courage to change the things I can, and the wisdom to know the difference. The Father, the Son, Holy Spirit. All right. Let's check this out. Got to get the projector going. Every layer of technology that you have to use takes an extra 15 minutes to get it to work. So let's see how this works. This is at least a half a minute to get the camera to come up. Is that working? Yes. So, while we are, oh, is it recording? Am I recording? Is it recording? Let's stop the screen share for right now. It is recording. Good. Okay. So we'll do a quick review of what we've been going over so far. We got the major areas of the brain, right? Because that's what we're really going to be focusing on. Close the door. I don't want you to go to the hallway. So we're talking about how drugs are mainly going to affect the nervous system. So major components of the nervous system. Hit me, Emily.
Central nervous system.
Central nervous system and peripheral nervous system. Okay. Peripheral nervous system is sort of divided into two big components.
Sympathetic and parasympathetic.
Okay, that's one. So we've got the central nervous system up here, right? What was that? Updating failed. Beautiful. Basically, we've got information coming to the central nervous system, right? And then we've got information going out of the central nervous system. So this branch is called the Donna. Right, this is operant. And this is all of your sensory information. And then over here we got efferent. Now in the efferent, we've got the autonomic nervous system, which is otherwise known as the involuntary, right? And in the autonomic nervous system, now we've got, right, sympathetic, parasympathetic. Over here, we've got somatic. You guys probably remember that. That's the voluntary. That's when I want to move. And then we've got the enteric nervous system with your guts. We're not going to talk too much about that. What's the main function of the central nervous system? Julie?
Good question. I don't know.
You're trying to wake up? Allison?
The processing and division making.
Processing, decision making, and then initiating responses, right?
Yes.
Good. So it's brain and spinal cord, central nervous system, and the brain is really where we're going to be looking at. We're really going to be studying major divisions, right? We've got the brain stem. Are we still in the brain stem? Yes, we are. We're still in the brain stem. And one of the things, hopefully, that's jumping out at you is that we've got major nuclei in the brainstem. So we have neurons, major nuclei that are synthesizing and releasing the real prominent neurotransmitters in the brain. A lot of these centers are located in the brainstem. So when we say we're affecting the brainstem, we're really going to be having a real profound effect on behavior. That should make some sense because the brainstem really is related to survival. So some of the things we're going to see, some of the behaviors that are going to be regulated by these nuclei, these pathways that are in the brainstem, a lot of them are going to be related to survival. Survival, okay? All right. But before we talk any more about that, I do want to take a look. This is a cool video. They're beginning to map out all the neurons in the brain. And by the way, most of the brain, if you guys read it all, that sounds bad. If you did some extra reading, most of the brain actually isn't made of neurons, is it? Most of the brain is comprised of the cell type. These different cells are called glial cells, glia. And glia means, Emily, you remember what glia means? It means glue. Well over 99% of your brain is actually not even neurons.
You said over 99%?
Over 99% of the cells in your brain are not neurons. There are these other kinds of cells, glial cells, which support the function of the neurons. So only 1% of your brain cells, okay, the cells. So, and one of the reasons for that is that neurons are pretty large. Glial cells typically are pretty small. So you can cram a lot more glial cells into a smaller space. So, anyways, so let's take a look at this. We're being able to map out now connections, networks of connections in this brain. These are all neurons. Stop. Okay. Did it finish? It did finish? Or did we get? Okay, all right. Making sure that the ad didn't cut off the video. So, and one of the other things that they're doing now, let's see here. I want to highlight. Stop. All right. So this would be in here. These would be major tracks that would be connecting the front, the anterior, and the posterior of the brain. Cortex and the fibula. We'll talk a little bit about these pathways. And then here, look at these. You see them going horizontally. So it's a structure called your corpus callosum, which really connects both the hemispheres of the brain. So a lot of what we see here, these are axons. A lot of these are neuronal axons. Right? The cell bodies are real small, so the vast majority of our brain really aren't cell bodies, it's these axons. And behavior is the result of the activation of specific networks of neurons. So you might look up some, that's what's called the connectome. So there's a lot of studies now where they're trying to see, using different colors, person is maybe depressed or anxious, or they're making a decision about performing a particular task, and they're then able to reconstruct which areas of the brain are activated, which areas of the brain are being inhibited in concert, which is kind of cool. So, anywho, all right, we'll get on with our regularly scheduled program. We finished talking about the midbrain. And why am I using my dry eraser? Devin?
Because your brain's telling me to.
Your brain's telling me to because I grabbed the wrong one.
Oh.
All right. So the midbrain. The midbrain. Let's see. Let me put you guys up in the corner here. The midbrain. All right, weird. I know it's not up here, but we were talking about the tegmentum. So the midbrain contains two major areas, right? The tectum and the tegmentum. Tectum is responsible for visual and auditory processing. Tegmentum, this is going to be more important for us. Contains a number of areas that we're really interested in. The first was the periaqueductal gray, right? That's pain. Moderates pain. Substantia nigra. With a question. First of all, substantia nigra. Helps coordinate or control, right? We said fine motor movements. What's the neurotransmitter here?
Dopamine.
Good. That's important. So you got a brain, so midbrain, substantia nigra, fine motor movements, dopamine. Link those together. That's important. Your question was, are they connected? They're not connected. So the cerebellum and the substantia nigra are not connected. The substantia nigra is part of a larger network that we're actually going to talk about today of brain regions that really coordinate. It provides a lot of, we would say, associative sort of information or supplemental information about motor movement. That group of, that region of brain areas is called the basal ganglia. Did you guys talk about that, basal ganglia? So both the basal ganglia and the cerebellum provide input to the motor cortex. So they're both sort of providing information. Okay, it's a great question. The third area that we talked about was the ventral tegmental area. And I'm simply going to abbreviate that as the VTA. Ventral. So when I hear that word ventral, for the brain, dorsal, ventral. This is dorsal, this is ventral. So ventral means it's down in the lower part of the brain. Tegmental, it's in the tegmentum, it's in the midbrain. Area, like you know what that is.
I have a question.
Yep.
So it's the substantia nigra that produces dopamine and not the ventral tegmental area?
They both do.
Okay.
They both do. So these two areas contain neurons that synthesize and release dopamine. So it's not as though, okay, yeah, that's a good question. So what happens? Let me switch over here. I just can't see the board. I think this will become more clear, Allison, whenever we start talking about the particular neurotransmitters. But what happens is, in the substantia nigra, that's a region where there are neuronal cell bodies, and all of those neurons synthesize and release dopamine. So the cell bodies of those neurons are found in the substantia nigra and the ventral tegmental area. And then what they do is the axons then, we call this projection, they extend to other regions in the brain. And the neurotransmitters are stored in those axon terminals. So let's come back to that when we talk about the neuron, which we should talk about today, okay?
Okay.
So that's a great question. Let's hold on to that and let's come back to that in a moment. Thank you. That's a good question. So neurons in both the substantia nigra and the ventral tegmental area synthesize dopamine, and then those neurons release dopamine in different areas of the brain. So in the VTA, there's two major pathways. So these pathways, their cell bodies both start, or their cell bodies are in the ventral tegmental area. But then when they extend their axons, they go to different areas of the brain. Right? So the first area, brain area we talked about, is called the mesolimbic pathway. And we'll talk more in detail about this. Right. Meso, so when you see a pathway, the first part of the word tells you where the cell bodies are, or where the origin of that pathway is. The second part of that name of the pathway tells you where those axons are projecting. So meso means it begins in the midbrain, that's where we are. Limbic, it extends to multiple brain nuclei that are part of what's called the limbic system. And this is considered, well, we're going to talk about the limbic system today, but the limbic system in general is considered to be, right, the emotional place in the brain. It's where our emotions are generated. And it releases dopamine. And all drugs of abuse result in the release of dopamine from neurons in this mesolimbic pathway. So if you do anything and your emotional message at the end of doing that, whatever that thing is, ooh, that felt good, or that tastes good, or that is just awesome. Dopamine being released in the limbic region. It's a signal. It's a chemical signal that tells you if you need to feel better in the future, do this. So that signal tells you. All right. The other one is called the meso. Does anyone remember? Yeah, Emily.
Mesocortical.
Mesocortical. So this pathway also releases dopamine in different cortical regions. This is going to the cortical regions. Important for cognition. All right.
How does the dopamine help with the cognition?
How does the dopamine help with the cognition?
Yeah.
So when it's released, well, when neurotransmitters are released, what we're going to sort of talk in general how neurotransmitter release sort of increases or decreases. It basically is going to increase or decrease the function of particular neurons in specific areas of the brain.
Okay. So basically, it just stimulates the parts of the brain for cognition?
Not, well, neurotransmitters don't always stimulate. Sometimes they inhibit.
Okay.
So it's the combination.
Okay.
So, yeah, that's a great question. And a lot of our information, a lot of the reasons why we we draw a conclusion about why a neurotransmitter release. Thank you. Why it has a particular effect on cognition, for example, is if you block a particular receptor, then cognitive function goes down. So then, well, I guess we need to activate this particular class of receptor with this neurotransmitter in order for the cognition to be able to happen, or memory, or learning. Okay. So, yeah, it is complicated. We're going to try to keep a simpler story. All right, that's a good question. Okay. Diencephalon. Moving on up. Moving on up. Contains the thalamus and the hypothalamus. Thalamus and the hypothalamus. Thalamus, it's this orange structure right here. We can see it's divided into a number of different regions. We're not going to really focus a lot on the thalamus, but it's important for you to know that all the sensory information that comes to the brain, before it goes to any of the sensory cortex areas to sort of begin processing, it goes through the thalamus. The thalamus organizes all that stuff, all that information. There's one sensory modality that does not go through the thalamus. That's your smell. It goes right to the sensory cortex. Right. The hypothalamus, well, you know, it regulates. The hypothalamus is right here. It's this green area. So hypo, below, thalamus, thalamus. So it's this green, these two little green dots. That's the hypothalamus. A lot of your internal functions are regulated by the hypothalamus. So it's receiving information about osmolarity of your extracellular fluid. It's receiving information about stress. It's receiving information about what time of day it is. And then certain hormones are released or inhibited depending on what the internal conditions of the organism are. And one hormone that is released by, its release is regulated by the hypothalamus is that stress hormone, cortisol. Everybody loves cortisol. Not really. So you think about the stress response, a lot of our stress responses are regulated by the activity of the hypothalamus. That's it for the mesencephalon. Okay, woo, we're gonna have to move. I'm gonna minimize these guys. So Allison, I'm gonna minimize you. If you have anything to say, you're gonna have to shout out, okay?
Okay, yeah, I have a quick question.
Not now. I'm just kidding. Go ahead.
Was
Yes, go ahead.
the diet, you said it was the mesencephalon, or was that the diencephalon that we just talked about?
The diencephalon, thalamus and hypothalamus.
Okay.
Yep.
So that's another, it's separate from the mesencephalon.
That's correct. We now moved out of the midbrain, so we're now out of the brainstem.
Okay.
We're no longer in the brainstem. We're out of that, and now we moved up. By the way, notice these yellow areas? I was talking about the basal nuclei that helps regulate some motor function. These are all these yellow areas right here.
Is the basal nuclei different than the basal ganglia?
They're the same.
Okay.
They're the same, yeah, just different names. Yep. Oh, and that's where we are. So, here we go. In the human, the cerebral hemispheres make up the largest region of the brain. All right. So we're going to talk about the whole telencephalon. We're going to talk about some different components of this. This is the cerebrum, sort of the top. You had an ice cream cone. One of the models of the brain is that everything we talked about before is kind of like the ice cream cone, and then you plop ice cream on top of it. It's kind of it's an okay analogy, but anyways, let's move on. So, the basal ganglia. Here we see the caudate. This is the caudate nucleus, the globus pallidus and the putamen. The caudate and the putamen are two areas that oftentimes are simply called the striatum. So these are just words, all right? These are just areas, but when you hear these, putamen, striatum, substantia nigra, here's the caudate and the putamen. Think this is going to be affecting motor function. That's like the main thing. So, do I have to memorize these areas? You don't have to memorize these brain areas. But if something's affecting the basal ganglia, be thinking this is affecting motor function. If I activate those basal ganglia, I'm probably going to see increased locomotor activity. And if I inhibit them, I might see depression. Just in big general pictures. Okay. Limbic system. The limbic system. It's all this stuff right here. That's a lot. Okay. This is a network of structures that, we've said this before, integrates emotional responses and regulates motivated behaviors. What do I mean by motivate? So let me repeat that. The limbic system is a network of nuclei or structures in the brain that integrates emotional responses and regulates motivated behaviors and motivated that are related to learning as well. So when I say motivated behaviors, what does that even mean to you guys? That means something. Motivated behaviors. Yeah, Devin.
It sounds like something your brain would get accustomed to doing over time and would bring you more happiness or more dopamine maybe?
Okay, so we're learning. Okay, what's motivated? So, okay, but you're learning.
It makes me feel like behaviors learned through like offering conditioning and reinforcement. It's something that you do and you learn that you get reinforcement, so you learn that it's good and then you motivate to do it more.
Okay, it's related to your happiness, sadness, punishment. That's also a motivator, right? I'm not going to do that again. What were you thinking, Emily?
Also not the same line.
Okay. Also, this is all good. Also has something to do with, like, how hard you're willing to work for something too. Right? So, Friedrich Nietzsche, the philosopher, the German philosopher, he said, anyone who has a sufficient why can endure any what? Anyone who has a sufficient why can endure any what, right? Is there enough meaning? How motivated am I to keep going through this? And that's all related to emotions, right? Kind of a drive you have to see things. So what are some things that you think you'd have just sort of an innate drive to seek? This is a biology class, so we can sort of, everything's sort of on the table.
Sex drive.
Okay, sex drive. Boom, limbic system. Next. Anything else? Hey Al, you trying to answer me or what? Why are you clicking? Yeah, Devin. Food. Okay, food. Anything else?
Something of like basic survival mechanisms, like, does it regulate like the fight or flight response?
Well, it's going to provide input to the hypothalamus about that. Especially, think about fear, amygdala, big one. Amygdala is big for fear in general. The hypothalamus, the hippocampus is more related to sort of learning, sort of helps with associating things. Do we have the nucleus accumbens in here? No, we do not. We notice that thalamus is part of the limbic system. We don't see the nucleus accumbens. That's a structure that you should know that that's part of this as well, nucleus accumbens. All right, sweet. So that's the telencephalon. No, that's not the telencephalon. That's the basal ganglia and the limbic system. Basal ganglia, movement. Limbic system, emotions, and motivational learning. Now we got to talk about the cortex, right? The outermost layer. The human cortex has these deep convolutions that are not found in other lower rodent mammals, such as rodents. So on the left, we've got our little rat brain here, nice and smooth. We don't see the convolutions. What's the whole point of having these convolutions?
It increases the surface area.
Increases the surface area. That's right. Increases the processing power in smaller space. Exactly. So this is just to give us a little comparison here. And we're gonna break the brain, the cortex up into these major structural areas. We're not going to get into all the associative areas. If we need to bring some of those out, so far as their functionality changes following the administration of a drug, we'll talk about those. But otherwise, we're gonna stick to the basics here. So the frontal lobe, this yellow area, right? Frontal lobe, the yellow area. It's primarily responsible for voluntary motor activity, speech, and what we call thought or cognition. So, voluntary motor control, that happens right here, this region. So this yellow region right here, this is the primary motor cortex. So it's the one that's right next to the blue one. So the yellow one that's anterior to the blue region, that's the primary motor cortex. And then there are also regions here that are responsible for speech. Speech tends to be on the side. And then thought or cognition, especially up in the front. frontal lobe. Let's move, let's go to blue. Let's go to blue, the parietal lobe. That's the blue area. The parietal lobe receives, synthesizes, and processes sensory information. So it receives and processes sensory information in general. And just as the most interior region of the frontal lobe is the motor cortex, the most anterior blue region, that's this region that's right next to the yellow region, right next to the frontal lobe, this region is the primary sensory cortex. Okay, so Johnny, right now you're picking at your right thumb. You feel that? Well, the reason that you feel that, one of the main reasons that you feel that, is that there's a region in the left hemisphere of your primary sensory cortex that's lighting up. It's actually connected directly to your right thumb.
I have a question.
Yo.
I'm just trying to make some connection. So with the parietal lobe, since it is dealing with synthesizing and processing and receiving sensory information, wouldn't that make it part of the peripheral nervous system, because your afferent and your efferent divisions are dealing with sensory information, well, especially the afferent divisions dealing with sensory and information?
Okay, so that's a good question. So what happens is, so let's get back to Jonna, when she's scratching her right thumb. That information, that stimulus, scratching the, why am I scratching my left thumb when I'm scratching? I'm saying right thumb and I'm scratching my left thumb anyway. So that sensory information, right thumb is being touched, that's being picked up by sensory receptors in the right thumb, and those receptors are connected to sensory neurons. That information then, that sensory information, in the form of an action potential, we'll talk about that, travels up the right arm all the way to the spinal cord. It then goes all the way up to the brainstem. That's one nerve. That terminates on a second nerve. Some on the right side. It then terminates on a second nerve. That second nerve then receives neurotransmitters. We get another action potential in that second nerve. It then crosses, which means it begins on the right side of the brain, the brainstem, flips over to the left side of the brainstem, and then goes up to the thalamus. And then there's a third nerve that goes from the thalamus, a specific region in the thalamus, that then extends to the primary sensory cortex in the left hemisphere of the parietal lobe. So by the time that information finally reaches to the sensory cortex, the nerve that actually relays or relates that particular sensory information begins in the thalamus, which is the brain, and extends to the sensory cortex, also the brain. So that final nerve is completely and entirely contained within the central nervous system.
But it begins as peripheral.
That's right, it's connected. But once it gets into the brain, it's no longer peripheral nervous system.
Right. That was my question. That makes a lot of sense.
Yep. Good. That's a good question, okay? Yep. Okay, other questions about that? And we're talking about these things in this is very big picture stuff. the minutiae, we're not going to get into a lot of minutiae about, there's a lot of different kinds of processing that happens at the areas where the different lobes come together. There's a lot of crosstalk between those areas. We're not going to talk about that. Occipital lobe in the back here. This is the posterior of the brain. This is the green area. Occipital lobe processes visual information. This is a part of the cortex, in addition to somatic, in addition to the sensory cortex, that also processes some visual information, occipital lobe. And then the temporal lobe, found here on the temples, sides, this is the pink area. What sensory organ is closest to the temples? Your ears. So guess what this region processes ultimately? Auditory information. All right.
Doesn't it also deal with smell too, or is that a different region?
I'm not sure about that.
Because I know there's like the olfactory bulb, which deals with the smell and taste,
That's
Right.
more, yeah.
but I wasn't sure if it did.
Did you guys cover smell in neuroscience?
We did, yes. I'm pretty sure it might be in the parietal lobe where it's that information process, but like I said, it's been.
So all that sensory information first goes to the primary sensory cortex, then it gets relayed to other areas.
Okay.
So that could be, I'd have to look that up. Yeah?
When you have hearing loss, what part, or like what in the temporal lobe, like causes that to?
Ooh. I don't know. That's a good question. Could you email me that question?
I'm not here right now, so I don't forget.
Sure. Last question.
The central sulcus, is that just one of the fissures in the brain, or is that like a separate like part?
Okay, good question. A sulcus is where you see the valleys. So a sulcus is a valley. A gyrus is sort of where you see the hills. So instead of hills and valleys, we have gyri, a gyrus, or a sulcus. Good question. All right, all good questions. Now, connecting this brain, this is just to give us some idea of what's actually happening. We saw this in the video with the connectome. Many of the neurons or the axons, a lot of the volume, let's say a lot of the volume in the brain is actually dedicated to the transfer of information, either from the front, which we call anterior, to the posterior, which occurs by a pathway that's called the fimbria, or between the right and the left hemisphere, either through the corpus callosum, which is this region here, and then there's another major pathway that connects the two hemispheres. One is called the anterior commissure, goes across the commissure, or the posterior commissure. So the whole point of this slide here is just to let you know that where you see the gray regions, that's where the cell bodies would be. So gray regions, if you're talking about gray matter in the brain or the spinal cord, that's where you have cell bodies. So most of this brain is actually not really cell bodies, is it? No, most of the brain is actually these vast networks of axons connecting parts of the brain.
So that's what the white matter does, it connects every organ.
Mhm. That is correct. Okay. Well, one of the ultimate goals of this class is actually to begin, near the end, when we begin to study, we'll study data from different studies. We'll cover data from different studies as we go through the semester. But ultimately, the reason that many behavioral studies are performed on rodents, rats or mice, is that there are many similarities between the two kinds of brains. We've got the human brain on top. We have our rat brain down below. So I just want to ask you just to take a few minutes, some moments, jot down some ideas, some thoughts about what similarities do you see between these two brains. Hopefully you can read some of these words. And then what are some of the differences that you might see or think about? So similarities and differences. That's important. Any thoughts? Any thoughts? Just some thoughts as we're just brainstorming here. We're brainstorming. We'll go with Emily, then we'll go with Emily. Emily.
I said some parts of the rat brain is larger relative to the size of the human brain. So like the olfactory bulb in the rat brain is really big compared to the size of the human brain.
Okay, so we're going to differences. Okay, so the ratio of the different brain structures of some, right, is different. Okay? How about some similarities? That's good, you jump in it, yeah.
I'll say that basic structures are similar. Like you have the brain stem, you have the cerebellum. All basic structure there is different stuff.
Right. So both the rat and the human brain have similar, actually they have all of these brain structures. They have them all. What about how they're sort of laid out?
The brainstem is pretty much the same, like the way it goes on the middle of spinal cord, the federal order. And it looks like it's pretty much the same size.
So we can say that the basic architecture of the brains is similar. They used to call, there was a very prevalent theory at one time where it said that the brainstem was the reptilian brain, and that some of these other structures evolved later. Well, they've done more research and they found that reptilian brains, they do have some of these other structures as well. So they're not exactly the same, but they have some similar functionality. I will also tell you that the functionality of each of these brain areas in the human and the rat brain is going to be very similar. By functionality, I mean, if I were to activate, I like the amygdala, if I were to activate the amygdala in the human brain, and we were to show you a bunch of pictures of faces. You guys cover these studies in psych? You guys study any of those? Where you get the smiling and
Yeah, that's like a social psych concept.
okay, where that you have to identify whether or not the face is happy, sad, negative, positive, whatever. Well, if we were to activate your amygdala, turn it on in a certain way, you guys, what you would probably report that a larger percentage of those faces were negative or angry or threatening than someone whose amygdala is sort of chill. Same thing with the rat. If you go in and you were to activate the amygdala, which we can do, we now have the ability to put light sensors, so light receptors in different regions of the brain, and then you just put a little cannula down, you insert a light cannula down into the brain, let's say it was the amygdala, and then you hit it with a bunch of bursts of light. You observe how the animal reacts. You would see increase in fear, et cetera, et cetera. You see the fear response, freezing response in the rodent. And they're sort of laid out the same. Okay, so this tells us, right there. This means that we have a high degree of what's called conservation, right? Conservation. We have a high degree of conservation of brain structures in mammals. Conservation, which means that if the brain region had a particular functionality, was beneficial to the survival of that organism, and then that got handed on, right? And then eventually we got it. So because of these similarities, this gives us some basic rationale for performing behavioral studies in rodents using psychotherapeutic drugs, and being able to interpret their results in light of the human being. Okay, we had some questions.
No, I was gonna add the similarity, but you already said it.
Oh, what's another similarity?
I was just gonna say that like the location of all of the parts of the brain are pretty much the same.
Sorry.
Yeah, you said it, it's okay.
And we have one more question? Yeah, Julie.
If you would stimulate the amygdala in a rat, is that how you could do, tests like on anxiety, like the rat?
You can certainly do that. So a lot of the earlier studies, they didn't stimulate those brain areas. You know what they did? They destroyed them. So they used what was called chemical ablation. And your book talks about this. Ablation, A-B-L-A-T-I-O-N. They would, there are chemicals that would, for example, would specifically destroy or, sort of block, if they could identify particular proteins, or there are actual, there are actually certain bacteria that migrate into particular kinds of neurons. So they can attach something that's sort of kind of destructive, or they can link something to that bacteria. They can then infuse that. It goes in, it finds the particular neuron, and then goes in and wipes out a bunch of nerves. Or they just infuse a chemical into, for example, we talked about a real simple one, the nucleus accumbens. We said that's important. I said that's important for the rewarding properties of drugs. Well, if you go in and you actually chemically destroy part of the nucleus accumbens, animal stops taking drugs. Doesn't take its cocaine anymore. It's not interested. So why do we do that in humans?
Because our nucleus accumbens is probably more complex than that of the rats. It would have more adverse effects other than getting rid of someone's addiction.
Okay. Maybe it's more complex. What are some other reasons why we don't just sort of just jump in and just eliminate something like that? Other than it's unethical.
It's slightly enough.
What were some of the surgeries that were done in the late 50s and early 60s to eliminate bad behavior?
You mean like a medical procedure?
Yeah,
Is
real
it
simple.
lobotomy?
The lobotomy, yeah. You totally annihilate the frontal lobe. It's really simple. Drill a couple holes in the front, put it in, scramble it up, brr, brr, frontal lobotomy. Look it up. We did it. We did it on people. One Flew Over the Cuckoo's Nest. You guys ever hear that movie? You ever see it? Kind of crazy, right? That's what they did to him at the end. You see the little lines on his forehead, I think, and he's just sitting there. So, another reason that we don't just simply chemically, let's say, suppress the nucleus accumbens, well, behaviorally speaking, what might suppression, chemical suppression, we're not going to say destruction, but chemical suppression of a region of your brain that's important for telling you if something is pleasurable or rewarding. What might, and we just shut it completely down. What might that look like in a human being?
Depression?
Yes, Allison, we call that severe depression. One of the symptoms of depression is something that's called anhedonia. Anhedonia. You guys ever heard of that term, anhedonia? What's it mean, Emily?
It means they don't take pleasure in the simplest things.
Yeah, so we've heard of hedonism, right? Hedonism. There it is, hedon. I think that's pleasure. Can't spell. And against. Yeah. I don't even know what that means. Condition. That's a condition in which you just don't care. You don't care about anything that's pleasurable. You won't even take a walk. You don't do anything that would be pleasurable. Okay? All right. Cool. Wow. Differences. How about some differences? We already got one. Emily got a good one here. That the ratio of certain brain structures is really different. What do we need? Point one out to me, please. Where we see a big difference.
The olfactory bulb
In
in the human in the brain, it takes up a big portion of the rat, but you can barely see it in the human.
okay, so the olfactory bulb in the rat is ginormous. Beep, it's teeny weeny in us. So what's that tell us about the importance of the sense of smell in a rat? Hi, up there. Texting?
Yeah, but it's like this one-on-one class, and something was due, and everybody missed it, so it's blowing up right now.
Okay, so in us, it's not as important, doesn't mean it's unimportant. What else? What are some other areas where we see big differences? Okay, Jordan?
The pituitary gland in the human is a lot smaller than rats. It's still small, but it's a lot smaller.
So, yeah. So it's in comparison to the size of the brain, it's smaller than in the rat. Good. What about Emily? You have your
The human cerebellum is like larger than the rat cerebellum.
Yeah. What do you think that is?
And it seems like we're larger than we have more complicated movements than a rat would.
Last time I checked. Yeah. Okay. We had Devin.
The cortex of the human brain is just far more intricate.
Boom. That's a big one, right? The size of the cortex. So the ratio, yeah, the cortex takes up the largest proportion of the brain in humans. And we already talked about how it doesn't have all the convolutions. Midbrain. Look how big the midbrain is. How much of the brain the midbrain takes up in the rat as opposed to the human. Okay, so there's a lot of these differences. So there are differences. That means that we can't just. That's one of the reasons. There's a lot of reasons, but that's one of the reasons why we can't simply say, I gave this drug to these rats, and this behavioral symptom was corrected. Now it's going to work in humans. One of the reasons that we can't just simply translate that information to the human picture is that there are differences. John?
I was gonna ask, it might be a dumb question, but is cerebellum also like smooth like the rest of it, or is it like more in depth like the human cerebellum? Because I don't know if the way that they just like made a diagram up or.
I don't think it's as intricate. So that's a good eyes you got there. So yeah, the human cerebellum, in addition to being larger, it's also a lot more, it has significantly larger surface area. The structure itself is yes, it's much more delicate, I guess. Yes. Cool. So, anyways, and the human brain, another difference is that the human brain, the rat cortex is going to have all those same basic processing function areas, right? There's a frontal cortex in the rat brain. There's a sensory cortex, there's a parietal cortex in the rat brain. Occipital lobe. There's going to be temporal lobes for general processing of that information. But the human brain is going to have much larger association areas where those are areas where different sensory modalities are brought together and processed, which is what helps us to have much higher ability or level of what we call abstract thinking. Right? We can actually think about an idea of a stop sign and what it means, or the color red and what it means. The mouse, you got to show it. Red, or light, or smell. For us, we can just think about a word, right? A word. Christmas. Boom. You probably had all kinds of things that pop into your brain. Why? Because we have these associated events. Okay. All right. Uh-oh, frozen. It's okay. Oh, it really. Uh-oh. Okay. All right. Spinal cord. Let's get through this. We're just. All right. Major things, spinal cord. Remember, this is also part of the central nervous system. It's continuous with the brain stem. So here's our brain stem, spinal cord, da 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 da. It extends from the brain stem all the way down to this region here in the lumbar back. And this is the posterior view, or the ventral view, and this is a side view, right? And so all these little white structures, these are all vertebrae. And extending out through the gaps between, the spaces between the vertebrae are going to be these spinal nerves. So you don't have to know all of the regions of the spinal cord. This is more for your information. It's not physiology. And so what we're gonna do now is we're gonna sort of take a cross section. So we're gonna cut through spinal cord, then we're gonna tilt it up, and we're gonna take a look at it. All right. This is what we see. So just to give us some orientation, ventral is back here, or dorsal, excuse me. Dorsal is back here. Dorsal would be your back. And the ventral part of the spinal cord would be in front of that. Easy way to remember that is, what's that process of when we breathe air in and out of our lungs? We call that ventilation, right? So ventilation is happening in the ventral area of your body. This is posterior, this is ventral. Or my brain, dorsal ventral, I'm sorry. So what I'm showing you here is that so these little nerves, see, this is a spinal nerve. There's going to be a pair of them, one going out to the right side of the body, one going out to the left side of the body. That's what we see here. So this is the left side of the body. This nerve contains sensory neurons that come into the spinal cord, and it contains motor neurons going back out to the body. All right, so these spinal nerves, we can sort of think of them as being like, a bundle of fiber optic wires, sort of organizing them and collecting them. So this is a spinal nerve. So a single spinal nerve contains both sensory information coming into the spinal cord, which then can go up to the brain, and also motor information. All right, this is just to give you some idea. This is just to show you how those spinal nerves extend out from the spinal cord. So now here, this is the dorsal region, this is the ventral region. This is your spine, these are your vertebrae. Okay. So that brings us to the peripheral nervous system. Julie?
So I know that in the spinal cord, the gray matter is on the inside as opposed to the brain where it's on the outside.
Yep.
Why are they different? Like, why wouldn't they be like one, they both on the outside or both on the inside?
That's a question that you need to ask someone on a bigger design. So I would say that if you damage, to here, this is the white matter, which means that this is actually axons that are either connecting the body with the brain or the brain with the body, right? So that information will be coming down, and the cell bodies would be in here. I've never really thought about that question, to be honest with you. Like, what's the practical reason why it's organized the way it is? So, I don't know. I never thought about that. So, it's the way that the cells migrated during development. It's ultimately why that ends up happening this way. But the whole, hey, Becky.
Did I understand? Like, basically, the question was how easy it is to damage the nerves in the spinal cord.
Well, they were asking. The question was why are the cell bodies inside instead of outside?
Well, I don't know about that part. I just went to a surgical demonstration about the spinal surgery, and like they basically could remove the bones and like not replace it with anything, and there wouldn't be any damage to the nerves still. So it's not that vulnerable of an area.
Okay. There you go. And the main thing is if you can damage the tracks, but thank you, Bertie. All right. So we'll pick up here on Thursday, and we're going to start diving into the neuron itself on Thursday. And geez, I guess we have a quiz coming up next week. What's that?
The 30th.
That's next week, right? Yes.
Yeah, Monday.
Yeah. So, all right. Awesome. Did you send me the email, by the way?
I did, yeah.
Good.
Question.
Hey, Allison.
So all the quizzes are done via Schoology, right?
Got it.
And is it open for like a certain period of time?
Yes. We'll go over that again on Thursday.
Okay. Thank you.
Yep. You're welcome.
Yeah, I just responded to the email you sent out today about bringing the package to class.
Okay.
That's what I responded to.
Oh, okay.
Because I for some reason, when I was putting your name in the email, it wasn't popping up. And I didn't want to type it all out, so I just responded to the email you just
We didn't even get there, so.
Yeah, that's okay.
You're all good.
Sorry for being on my phone in class. I just was really panicking about not submitting the assignment that was due in my other class.
So if that comes up, what you can do is you can just maybe just pop up and go out, take care of something,
Okay.
and then come back in.
All right, thank you.
Yeah.
I didn't mean to seem rude. It was like in the heat of the moment.
No, we're in free fall, you know?
Yeah.
So, yeah, I get it.
All right, thank you for being understanding, but
Yeah.
I won't let it happen again.
Oh, that's okay.